ready, I want to introduce him again. Dr. John Halfman, Professor and Chair of Environmental Studies, the Finger Lakes Institute at Hobart and William Smith Colleges. He has been test sampling the water here for over 15 years in the Owasco Lake, and we anticipate his annual report and perspective and his experience in what's happening to the lake and what he predicts for the future. So take it away, Dr. John. Thank you for being here again today.
Thanks for that good introduction, Rick. Hopefully everyone can see my screen. Yep, okay, that's good. Hopefully everybody can hear me. Maybe you don't want to hear me, whatever. All right, so here's my 10th, I guess it's my 10th edition of the status of Owasco Lake, focusing mostly on the data that I collected in last summer in 2021. Here's a good aerial shot of Owasco Lake taken by drone a couple of years ago, and some other aerial shots around the margin showing some blue-green algae blooms that have been impacting Owasco Lake for 3, 4, or 5, I guess it's now 6 or 7 years now. So this for me all started back in 2005 when students of mine went to 8 of the Finger Lakes and looked at water quality indicators, trying to then rank things like bacterial counts and algal concentrations, various nutrient concentrations, suspended sediments, and water clarity, and then basically came up with a qualitative trend where the best water quality down here is where they seem to have the best water quality protection legislation, and the worst water quality lakes up in the upper right-hand corner seem to have the least water quality protection. And then if you then focus on which one's in the upper right, it turns out to be Owasco. And then you show this information to people like some of our former state senators. They said, okay, well, perhaps we should give you some money to then try to figure out what's going on. So ever since then, my students and I have been going on and trying to figure it out. The first year with funds from Senator Nozzolio, basically there was a huge number of sites going from the north end of the lake down to the south end of the lake. There was a cluster of them down near the Owasco Inlet, which is the biggest stream coming into the system. And that first year, 2006, there was some very high total phosphorus concentrations. The DEC considers 20 micrograms per liter here as the threshold for water impairment, and a number of these sites were significantly over 20 micrograms per liter. And it turns out that those sites that were over were down here sampling blooms coming in from the Owasco Inlet. Hopefully you can see my mouse down here. When you sampled the brown, you got lots of total phosphorus. When you sampled the blue water, you got back to normal lake values. This was also concerning. That then allowed me to get some more money to come back the following year. In 2007, I came back and looked at exactly the same number of sites. Noticed the southern end didn't have any impairment at all. And I was shitting bricks and wondering what was going on. And basically the difference between the 2 years, 2006 had lots of rain. So there was lots of opportunity to sample plumes coming in from runoff, whereas 2007 had minimal amount of rain, and the amount of runoff bringing in total phosphorus was minimized. How all this is going to impact a typical lake? Well, if you bring in nutrients into the water column, plankton can take up those nutrients. Once the plankton start to grow, it then feeds the rest of the ecosystem. When these alive things die, bacteria can decompose that material, releasing nutrients back into the water column, which then feeds more plankton. So you have this continuous cycle, a nutrient cycle. If you then change this nutrient cycle and perturb it by bringing in more nutrients, whether it's from agricultural sources or wastewater treatment plants, or more particulate matter from CAFO operations or human waste and soil particles, the bacteria really doesn't care where all these nutrients or organic matter is coming from. They'll still make more nutrients, which can then make more plankton, which then can make more fish and other organisms everywhere else. Notice that all these boxes get a little bit bigger. All of them get a little bit bigger. So they basically distribute what's going on. So it's hard to really detect if the nutrient concentration changed significantly, because when there's more nutrients, it goes to plankton, then it goes to other organisms. They all die, eventually gets down into the sediments where bacteria can then decompose them and put everything back. Some of the implications: you can get more weeds in the nearshore location, weeds like macrophytes and other things. You can get more cyanobacteria. If this was to extend too far, like as in the case of a Tisco Lake over to your east, or Honeoye Lake over to your west, the bottom waters can turn anoxic. You can start then making less pleasant smelling gases from the bacterial decomposition, and it's not necessarily a lake that you really want to be around. So my 2021 effort had two main things. There was lake and stream monitoring, which consisted of going out to 2 lake sites on a monthly basis, and then once the HAB season came around, it increased to a weekly basis. It also includes sampling some streams, primarily Dutch Hollow Brook, but also the Owasco Inlet and a couple of smaller tributaries on the western side. Then the other main aspect was deploying a buoy from April or May through November to collect daily water quality profiles and meteorological information, and then putting dockside sensor arrays at 4 sites around the lake, which complements what I'm doing in Seneca Lake, to look at what triggers or what meteorological and limnological factors trigger blue-green algae blooms. All this has been going on now for a number of years, and I seem to be getting a consistent story from all of this. So as I hinted at the very beginning of this talk, rainfall seems to be a big driver for water quality in the lake. It turns out that 2021, last summer, was a wet year, especially during the summer and into the fall. The spring, not so wet, but those 2 other seasons were significantly wetter, especially was significantly wetter than 2021, the previous year, when things seemed to really dry up. This one is on par with some other wet years, a little bit less than perhaps 2011. That number will come back to haunt us here when we look at nutrient loading through time later on in the talk. So if you look at what happened in 2021, there was a huge rainfall event that I'm sure everybody remembers that lives on the lake back in August 18th going through August 20th. This huge event, here's a rainfall depiction of it, of at least the first 2 days of that event. Notice here in red is rainfall accumulations. The largest ones are down here in red, whereas everywhere else it was relatively mild. It suggests that Owasco Lake really took a big hit. Small, localized thing. It's something that's going to get more and more prevalent, small, localized events due to global warming. And what it did to Owasco Lake is, here is one of the sites that I actually put my dock sensor on, that you can actually see the weather station here at this person's dock. This is what happened after the rainfall. Lake levels went up a little bit. As they went up, it released more nutrients into the water column. There was a significant bloom, but that's a later part of the talk, and we'll get back to it. What it did to Dutch Hollow Brook, the main second largest tributary in the watershed, the one that I monitor very extensively, is it's normally down here. But after those 2-3 days of rain, it got up to here during flood height. High enough to then take my auto sampler, which is sitting over here on relatively high ground, and just whisk it down the river. And volunteers found it floating a couple of miles down the lake. It ripped out a number of my data loggers and a bunch of other things, which were attached to 150-pound weights. I don't know, it was a relatively significant event. What this is related to is a recent climatic phenomena. They're called atmospheric rivers, which can then provide an avenue to bring moisture up from the Gulf and along the western part of the Atlantic Ocean all the way up into the Northeast. And it then provides and dumps huge localized events that might happen one year, might happen 2 or 3 years later, but it will happen somewhere throughout the Northeast, depending on the meteorological conditions up here as we get further north. This is something that one of my colleagues is looking at, Nick Metz, and this diagram's from one of his recent papers that's currently in press. What happened to Owasco lake levels? Here we have lake levels going back from 2011 all the way up through 2021. There's the August event right there, where lake levels increased by approximately 2 feet, maybe more. If it starts down there, it's a little over 2 feet. You might try blaming the outlet and they didn't let enough water out through the outlet, but in my opinion, they had everything full bore. Matter of fact, if they let out too much water, everything downstream is going to flood. So it's a very delicate balancing act of what to do, especially when you get a really big event in such a small amount of time. Gets aggravated in the Owasco watershed because the watershed is huge compared to the lake volume. So you get huge lake levels here. Don't blame West and his group. He's doing a great job. So for the stream monitoring effort, as I mentioned before, it's focusing on Dutch Hollow Brook. It's this thing in green over here. There's another sample site down here sampling the Owasco Inlet. And there was 2 more sample sites sampling small tributaries over here on the western side. I just noticed this morning that I forgot to flag fire lane 26. Sorry guys, but that's just the way it is. Things that I measured was discharge. There was samples for nutrient concentrations and a few other things that we'll talk about here in a little bit. If you look at discharge flow rates, basically it's very typical of a normal year, much higher discharges than a dry year. And if it was really, really wet, a little bit less than a really wet year. If you look at mean daily discharge through the months of April through October at the various sites that I've always continued to look at, 1 year back here in 2021 was high. Here we are, and excuse me, 2011. Here we are in 2021. It's a little bit higher, both in the Owasco Inlet and also down here at Dutch Hollow Brook. You look at nutrient concentrations in terms of total phosphorus and dissolved phosphorus. This is actually soluble reactive phosphorus. We're slightly smaller than previous years, but my grab samples were focused in May and June when rainfall rates were relatively low. So it's consistent with lower rainfall events. And if I had included some grab samples during later in the year, these values would have increased significantly. If you look at nutrient fluxes, where a flux of nutrient is actually the amount of the material going down the stream, and you determine fluxes by multiplying the concentration times the discharge, these fluxes are very similar based on the samples that I collected to a regular normal year. Whether you're dealing with total phosphorus here, here's a previous year for comparison where the numbers are a lot smaller because it was a drier year. Here's soluble reactive phosphorus. And then looking at some other inputs of phosphorus that are known, here's the inputs from the wastewater treatment facilities down in the southern end of the lake. Notice that you go down here to the Moravia wastewater treatment plant, it's always been low, below what the DEC considers its limit, that do not exceed type of limit. Whereas the one up in Groton was relatively high before the DEC came on board and then told them during this transition right here to OSD bound down below a similar limit. So those 2 wastewater treatment plants have been doing, in my opinion, reasonably well at trying to reduce the amount of nutrients, specifically the amount of phosphorus that's getting into the lake. Oops, go down again. Then Dutch Hollow Brook, I had some daily samples to try to figure out the differences between event and base flow. Here's an event going down Dutch Hollow Brook after a heavy rain, not the heaviest one that we saw last summer, but a relatively heavy rain. Here we are in base flow. Notice there's a considerable difference. Really muddy. The stream is full of nutrients, really clear, not full of nutrients. If you look at just the discharge going down the stream, you have an event, you have a rainfall event and the stream goes high, and then it slowly recedes back down to base flow conditions. And then you have another event, the stream gets high and you slowly go down to base flow conditions again. Then you had this event back in August 18th and 19th. It took away my data logger, which was unfortunate, but luckily there was a hi-fi sensor measuring stream heights at the same time. So I was able to use those values to interpolate a good response, where I got some of the highest levels in this brook ever measured in my 10 or so years of sampling sampling the stream. You measure concentrations during these times. Whenever there's one of these events, concentrations go sky high. I had to estimate the biggest ones because my sampler got lost both here in August and also in October. But even these estimates, the amount of concentrations go sky high. So if you total up all of these events and you look at the events versus space flow, the total load of total suspended sediments in 2021 was really high. It's one of the highest on records in the past 10 years. You look at the total phosphorus loading down this stream, it's the highest that's on record over the past 11 years. And in both cases, over 95% of the material came during events, during rainfall events. So in my mind, if you want to stop the loading of phosphorus and suspended sediments and other nutrients coming into the lake, you have to somehow curtail the phosphorus coming up from events. Sounds simple. Actually, in practice, it's probably not quite that simple. So if you then try to use the few data points that I got to try to figure out a phosphorus budget, in other words, using all the data that I've collected to get a stream input, you determine that the atmospheric input's relatively minor. You go out and you can figure out what's in the lake and what goes out the outlet. A little bit then goes down through and gets buried into the mud. You then balance all these inputs and outputs, and then you come up with a graph that's shown down here. Notice back here in 2021, that really wet year, lots of material was coming in compared to what was going out. The orange bars here is what's coming in, whereas the blue bars for each year is what's going out through the outlet. As you work your way from 21 all the way up through to 2016, almost every year more was coming in than going out, suggesting that you were adding nutrients to the lake and making things greener. There are some remediation efforts that were going on throughout this part of the decade. Notice that some of these inputs started to go down. This is good. If you look at my estimates of inputs and outputs, it's very similar to what the SWAP model predicted. SWAP model that's being used for the 9E plan that's being developed for the Owasco watershed. And then we come up into 2021. If you take everything but that one huge event in August, it looks like a year that's very similar to some of the ones in the past. You add that 1 year or you add that one rainfall event that occurred in August 18 and 19, it added approximately 50% of the load. One event, 50% of the load. And it's events like these that are in theory supposed to get worse into the future as we get into global warming scenarios. This to me is disappointing. We need to do more to curtail phosphorus inputs coming into the lake if we're going to combat events like this that are going to become more frequent into the future. You can then look at all this by source. Basically, I can divide up everything coming in by streams and then add to it wastewater treatment plants, an estimate from shoreline septic systems, and the estimate coming in from the atmosphere. And this is what you come up with. The blue is human waste. Either it's municipal wastewater treatment facilities or shoreline septics. In various shades of orange and yellow is all the other tributaries, Dutch Hollow Brook, and down here is the Owasco Inlet. In green at the very top is the atmospheric input. Notice through every year I come through, it's dominated by fluvial sources. Fluvial sources are primarily non-point sources. Which non-point sources are difficult to say based on the data that I collect, but that's just the way it is. Most of it's coming in by streams. So if I had to direct lots of limited money trying to reduce the amount of nutrients coming into the lake, I would then work on these non-point sources that are coming into our tributaries, both the little ones and the big ones, and try to curtail phosphorus that way. I'm pleased that my shoreline septic estimates are very close to what Nick Colas came up with his GIS models in the past year or so. He's been working on this to put in the information into the 9E plan, so that what you see here in blue is very similar to what he came up with his independent modeling efforts as well. Based on the 9E plan, they had a SWAT model that tried to then model phosphorus and other nutrient inputs into the lake. The SWAT model uses things like land use character, it uses rainfall history information and steepness of slope and other things to then predict how much phosphorus is coming in from different size events. So if you look at the basic input into the model, it looks at forested land shown here in green, and agricultural land that's in the watershed shown here in yellow. All the other land uses are up here, and urban is shown in orange, and then there's a few others that aren't even shown. So we're dominated 50-50 agricultural land and forested land. And then you put some rain on that water and you figure out what the total phosphorus loads are going to be. And it turns out that 90% comes from agricultural land. On a per acre basis, agricultural land provides more phosphorus into a nearby stream than forested land and other land use areas. It's split between cultivated crops and pasture hay, where a lot of the pasture hay land is places where you spread manure from CAFO operations. The cultivated crops also get some manure, but also get some fertilizer to augment crop yields and other things along those lines. So it's a SWOT model that can then help me pinpoint the sources a little bit better than I can, help me pinpoint the non-point sources and say, okay, it's primarily in the agricultural sector. So if I was to then distribute money, whatever available money there is, I would use this data and say, hey, look, we got an issue. It's impacting the lake because we have cyanobacteria blooms and other detrimental impacts in the lake. It's hurting our economy because we're basically ecosystem type economy. Let's give all the available money into the agricultural sector to then allow them to then reduce their nutrient loads and hopefully make a healthier lake down the line. This SWOT model has its advantages in that you can then model various strategies to reduce total phosphorus inputs into a lake. The model found that 3 of them were very, very useful. One was extensively use winter cover crops. I noticed that a lot of farmers are actually doing it. Maybe more farmers can do it with a little bit of extra funds coming into the system. All farmers, this plan suggested that all farmers follow nutrient management plans so that they know where the nutrients going, and then try to restrict manure and fertilizer use or spreading onto the land. If these 3 things are followed, you will then reduce phosphorus loading by about 70%. So 3 things to do through all agricultural activities, reduce phosphorus by 70%. This would be really, really cool. And this is what the advantage you have of using a SWOT model. I was also pleased to hear that Cayuga County is planning to continue the SWOT model into the future, collecting more data, more tributary input data to then refine the model. And it will be very useful to determine if some of these remediation strategies actually work in terms of the SWOT model and the actual data that you can collect from streams. So curtail degradation, since most of it's coming in from rainfall events, clearly you got to somehow reduce the amount of phosphorus getting into runoff, the amount of phosphorus that's getting into turbid plumes like this one coming in through the Owasco Inlet. Good ways to do it is to look at the revised rules and regulations, which is working its way through Albany as we speak. Complete them and follow the recommendations that are coming out in this 9E plan. Both of these have ways to then stimulate money coming into the watershed to be able to do it. So the financial burden doesn't have to be solely on the farmers. Clearly, we got to work on the agricultural system, put best management practices in place to curtail runoff events, have a little bit more responsible manure spreading. I've always advocated trying to send the manure from cows into wastewater treatment facilities, but that's a very expensive undertaking. It's not millions, it's probably in the billions of dollars range. There are cheaper ways just to remove the phosphorus from these animal wastes before you spread it someplace. Bioreactors is a good way to do it. Another source that you might try looking at is the amount of phosphorus coming in from drainage tile that drain agricultural fields, and the amount of stuff coming in from roadside ditches. Both of those could be done remediated relatively easily, in my opinion. Going to lake monitoring, see if this huge rainfall event had something to do with water quality in the lake. I've routinely measured sites 1 and 2 since day 1, 2005. I also have recently put in a buoy at this site right here in the middle of the lake. At all my sites, I'll deploy a CTD cast, which measures temperature, conductivity, DO, pH, the amount of algae and turbidity, also the amount of light available. Secudus depths and plankton toast. We do a little bit of spectral signatures of the water to try to figure out the spectral signature of blue-green algae blooms, so that it could be done by drone later on. And then I collect surface and bottom water samples for routine analyses, including nutrient analyses, total phosphorus, SRP, soluble reactive phosphate, nitrates, chlorophyll, and other things. Here we have secudus depths. If you're not familiar with a secudus, it's this disc here. You lower it down through the water column until you can't see it. That depth is an indicator of water clarity. The shallower the depth, the more algae in the water. And what's interesting is, looks like 2021 got a little bit deeper on average than previous years, suggesting the water got a little bit clearer. Water quality improvement, or perhaps something else. If you go to the next slide and look at the CTD profiles, here we have temperature profiles from all the days that I went out last year. And here's the algal concentrations. Notice almost all the algae is down here at depth, much deeper than what the secudus depth would then look at, suggesting that something's going on here at depth to promote algal concentrations to really flourish. And that something is, if you look at turbidity profiles, there's a lot of turbidity down here. And essentially what's going on is, when there's a big rainfall event, most of that turbidity gets into the lake and flows to the north end of the lake right above this thermocline. And if you have all those nutrients down here right above the thermocline, there's sufficient light down there. So you will actually end up with a lot of algae down right above the thermocline as well. Mist at the surface, but down here at depth. And last year happened to be a major impact on water quality for the entire lake, not just the surface of the lake. Here we have total phosphorus concentrations. Notice 2021 was relatively high, reflecting all the runoff coming in. You go over here, chlorophyll concentrations went down a little bit in 2021. Well, it went down because the algae was deeper in the water column than the surface water grab would then measure. If you can then combine and look at the trophic status of the lake by combining secudus depths, mean values, nitrate concentrations, total phosphorus concentrations, chlorophyll A, the amount of oxygen in the water column, you end up with 3 indicators showing mesotrophic lake a little bit more productive, 2 indicators being an oligotrophic lake a little bit less productive. So it's still borderline mesotrophic oligotrophic lake. You look at the plankton, just based on plankton counts, 2011 had lots of cyanobacteria, shown here in black, compared to other algal groups. Very similar to 2021, which is a lot more than previous years that I've been looking at the lake. If you look at fluoroprobe analyses of the surface water, which measures algae by algal groups by different pigments, when cyanobacteria was in the water column was down here at the later part, going through August into the end of September. Just like what happened here in 2021 and also in the previous year. A little bit concerning for me is this upward trend. A lot more black, a lot more cyanobacteria in the past 3 years than what you ever saw in the previous years. So things are getting a little bit worse in that respect. You look at cyanobacteria in a little bit more detail. Here's the locations where they formed, or where the local watershed group found blooms through their surveys. Few more blooms along this western shore. I think it's related to the fact that the dominant wind direction shifted from primarily south to south-southwest, allowing onshore winds along this western shore to then promote have blooms along that western shore. Here's a bunch of earlier dates shown up here in the upper right. Notice that the number of blooms that were found, detected, went down a little bit from 2021. Why 2021 was so high, I have no idea. If someone can figure that out for me, let me know, because I'm trying to write a scientific manuscript on the whole blue-green algae scenario as we speak. So what makes them tick? Or what makes a trigger for blue-green algae? Well, first I like to point out that if you go from 2005 all the way up to 2018 in all the various lakes that I sample throughout the Finger Lake system, we've always had blue-green algae, always. Matter of fact, you go back to some of the earliest limnological surveys in the early 1990s, blue-green algae. However, they didn't bloom, and they didn't bloom until recently. Maybe the past 6 years or so in Owasco Lake, which is here, other start years for some of the other Finger Lakes. So what triggered these recent blooms? So you look at what they like. They like warm water, they like sunny skies, calm conditions, and they like excessive nutrients. Well, this last one is a misnomer because Finger Lakes are nutrient poor, but let's look at some of the other two and see what some of my data can show. So here's Owasco Lake surface water temperatures recorded by CTD, starting in 2005, not so many sites, and then get into where there's buoy data here in blue and also superimposed with the CTD data. There's this warming trend, so warmer water, that makes sense. Then you compare it to the dock locations. This is the actual temperatures, a histogram of the actual temperatures of when I detected blooms. Notice they like warmer water, but they also liked colder water. This is new. This is interesting. This is confusing. I'm trying to figure it out as we speak. And there's reasons for why it might happen. If you look at some of this other data that I've been compiling over the last 3 or 4 years, over here we have wind speed. Almost all the blooms shown here in red happened at wind speeds below 0.5 miles per hour. Or 1 mile per hour. Really cool. The next thing, it should be sunny skies. Here's sunny skies right here. It's not the dominant peak for what's going on. This is a Seneca Lake graph, but exactly the same things happen for Owasco Lake. The majority of them come here in low light conditions. Another confusing aspect of, you should never have a lot of good data so you can plot it up in different ways and make everybody, or make myself confused. But it's something that has to get explained here as well. Air temperature, same pattern for blooms as non-blooms, shown by in the gray bars behind. Cooler water, we already talked about. When blooms happened, it's rarely raining. That makes sense. Rain brings cloudy skies, they don't come. When it's not raining, you get a lot of blooms. Their date over the past 3 years, sometime in the mid-August, late August timeframe, they have a huge outbreak of blooms, and then they trickle on throughout the rest of the year. Looking at whether it's an onshore breeze or an offshore breeze, in blue here is onshore breezes. Looks like they can withstand onshore breezes, but they can't withstand breezes that are going offshore. Now, I would have thought that onshore breezes would have mixed them into the water column, but apparently not. Apparently they can survive. Now, some of these blooms at higher wind speeds are at dock locations that are a lot better protected than I had hoped, so that you can have a strong wind, but the configuration of the land is such that there wasn't any waves right at the site where I was measuring, so you can have these excess blooms at higher wind speeds. But notice the majority of them happened at very low speeds, as shown in the previous graph. Nutrient concentrations. Well, you can estimate the amount of nutrients in the blue-green algae concentrations that were measured back in 16, 17, and 18. And from that, you get a number of how much phosphorus must be in those blooms. And then you compare it to the amount of phosphorus that's out in the open water. No matter where I look, if it's at the docks, just offshore of the docks, or out in the open water, always the concentrations are way lower than what you need to then support some of the big blooms that we've been having in the last 4 or 5 years. Always. Here's a bunch of nearshore sites. Here's the offshore site data. It's relatively disappointing. And it turns out probably what's going to solve what makes blooms tick is trying to figure out what this nutrient source actually is. So you set up a bunch of stations, a camera that can then document when there's a bloom, a weather station that measures wind speed, direction, light intensity, rainfall, barometric pressure, air temperature, blah, blah, blah. You want it, you get it. A couple of sensors that are in the water column. One's a water quality sonde, in other words, just measuring the amount of water temperature here. And then you put all this stuff together. Part of this work is also going to start looking at the organic matter in the sediments and the microphyte and mussel surveys along some of these nearshore locations, and also including taking pictures by drone and then getting a spectrophotometer, a detailed sensor on the drone to then try to figure out or identify drones from the air. That would be really cool because drones are really fun to fly. So you look at some of this information. Here's the information from the cameras. You can really see when there's a bloom, and sometimes when there's a bloom, you can see that the longshore currents are actually moving, transporting these blooms from your house to your neighbor's house, and hopefully to the neighbor's house beyond that. If you look at the time of day, seems to be relatively in the afternoon hours when they're coming. The number of blooms for the northern sites is relatively higher than what we found down in the southern part of the lake. You look at blooms duration. When there is a bloom, it's anywhere from 3 to 4 hours, but some of them extended for up to 12 hours, and some of them only lasted for about 10 minutes. So there's a huge variability there. Wind speeds are I didn't plot 2021 because it's the same story and it would have made a busy graph, but any shoreline site has slower wind speeds than out in the open water where the buoy is at. Slower speeds mean more common conditions. More common conditions means the fact that you can then have blooms. And if you look at the wind directions, here's the dominant wind direction out in the middle of the lake. Notice the wind directions for these sites are completely different. And it suggests that you can have blooms at one site because the wind's blowing offshore, but then no blooms at another site because the wind's blowing onshore. So it helps explain some of the variability in bloom locations when they do arrive. If you look at the percent calm conditions down here, the buoy rarely had calm conditions, whereas these nearshore sites had a significant percentage of calm conditions, most of them occurring at night, but significant percentage of calm conditions. Turns out that huge rainfall events, really huge rainfall events, like this one right here, made lots of blooms, triggered lots of blooms. And here's the map of the rainfall intensity. Yet once again, notice it impacted Owasco Lake. I had all these dock sensors around Seneca Lake, no impact whatsoever. This typifies the localized nature of the rainfall event that we had last year and will probably get into the years into the future. Then we get into the water quality sondes. Something that happened last year and was duplicated this year is at Burtis Point, the water quality sonde started to go down to anoxia for the dissolved oxygen concentrations. I should back up. There's a daily oscillation going from high to low, high to low. The high concentrations are in the daytime, the low concentrations are at night, suggesting that photosynthesis during the day makes the peaks, whereas respiration during the night makes the valleys. And there's a huge fluctuation, suggesting that there's a huge input of biological activity at these nearshore sites. Then you had some anoxia events. And there's one down here later on in October that was pretty significant. What happened during these events is inside the deployment pipe, there was, I had a PVC pipe and put this sonde inside the PVC pipe. Inside that pipe, vegetation got ripped up and encased in the pipe, making a microenvironment that then didn't get flushed out very well. So any bacterial respiration could then deplete the amount of oxygen inside the pipe. And as you deplete the oxygen by decomposing organic matter, you're going to then generate nutrients inside that pipe. If you then compare it to the amount of chlorophyll, the amount of chlorophyll shown here in green on this scale over here, which is much bigger than the cyanobacteria chlorophyll shown in red, you started releasing from those macrophytes some of the cyanobacteria, excuse me, some of the chlorophyll from the macrophytes, which then get decomposed. And as they get decomposed, then you have the nutrient source inside that pipe to then make a huge blue-green algae bloom within that pipe. Couldn't see it outside the pipe. Notice the cameras didn't detect it, which was right next to where the pipe was deployed, but you could see it inside the pipe. It suggests that if you rip up a bunch of macrophytes, you then rot them away using bacteria, release those nutrients. That there might be one of the nutrient sources which can then trigger a blue-green algae event along the nearshore setting. Makes sense to me because when you rip up macrophytes, they then blow along the shoreline and they stop right at the shoreline where they pile up and then start to rot. And then you get waves lapping into these piles of macrophytes, and it will then release the nutrients back into the water column to then allow the precipitation of a bloom. If you don't think it happens, well, I did a couple of experiments. One is I took flasks, I put some mud from Owasco Lake in the bottom of the flasks, I put distilled water above that mud, I swirled the flask, typifying at some sort of wind event, and then within a week, every one of these flasks had a blue-green algae bloom. Now that distilled water I first put in had no nutrients. So the only place you could have gotten the nutrients and the only place you could have gotten the blue-green algae was from the mud. Blue-green algae do make a resting stage, which sitting down here in the mud, so potentially it then might work. And then along Seneca Lake, you have this big mass of weeds that are accumulating along this shoreline. You could actually see the blue-green algae, the cyanobacteria, oozing out of this mat as the waves lapped up onto it and generate a big bloom. Places where I didn't see weeds accumulated along the lake shore, rarely did I see a cyanobacteria bloom. So maybe there's a correlation between the two. So for HABs remediation, perhaps you might want to use onshore bubblers or ultrasonic devices. Neither one of them seemed to work really well when we tested them in Owasco Lake a few years ago. Might want to put benthic mats down to prevent the growth of weeds and perhaps the migration of the resting cell for cyanobacteria into the water column. In contrast, I would never recommend using herbicides because your water, your lake water is used for drinking water, and it's the supply for up to 50,000 people. So don't put herbicides there. You don't want to kill off your population in Auburn and Owasco and other places. You might try removing more and more of this nearshore organic matter, which the macrophytes or the twigs or the leaves and other things, and stick them into a landfill someplace or put them in the back of your property at a minimum, because then you're removing that nutrient source from the water column. Because if you don't, some of this ancillary information suggests that bacterial decomposition of this organic matter will then release nutrients to then make a bloom in the nearshore area. We're working on zebra and mussel counts to see if that has an impact and might even get into Asian clams. But the bottom line here, out of all these things, is what triggers blooms is getting excess amount of nutrients into the water column, or at least into the sediments where the cyanobacteria resting cells are located. The ultimate way to do that is to reduce nutrient loading in the lake. So I think with that, I'm going to end because I'm getting near the end of my time, and I'm willing to take questions from people who are in the audience. So what I'll do is I'll stop my share so that we can get back to audience members here and go from there.
Dr. John, thank you. Rick here. I have a few questions or comments. During Tropical Storm Fred, they dropped warmer water and the surface water rose a degree or two in August. Did you observe something similar in Owasco Lake?
Yes.
Okay. Are base flows as large and as cool as they were 50 years ago? What is the impact of removing trees along streams that shade the water?
I don't know 50 years ago because I was a little kid then. But as far as the rainwater tends to be a little bit cooler than base flow. But it depends on the time of year. And it depends on the time of storm. So it's actually a mixed signal.
Okay. There seems to be some research toward feeding cattle seaweed, which has apparently a significant reduction in methane and related waste. Are there alternative feedstocks that you know of that can help in that regard?
You're treading on part of my expertise,
Okay.
which clearly is not there.
Okay, well, well, you just talked about not adding chemicals to the water.
Right.
And there was a comment that the earliest blue-green algae date was around 1912. I think you mentioned something, the first
Yep.
early blue-green algae data.
Yep.
One long one, can you use machine learning or AI to set meteorological parameters like wind speed, direction, rain amount, cloud cover, etc., along with your dock-mounted sensors to forecast and follow in real time and have proactive alerts as come to you as a monitoring researcher to anticipate location and extent of HABs, I guess?
All right, so that sort of thing, using some of these comparisons, in other words, blooms like calm water. Well, it turns out that when you don't have blooms, it also is dominated by calm water. So you can't say during a calm day you're going to have a bloom. The other impact that's here is you might have a bloom on one side of the lake during a calm day, but not the other side of the lake. So there's something else that's going on. You actually need a sequence of events, and the biggest sequence that I've been able to find is you start with either wind or rain that then mixes the water column a little bit, and especially in the nearshore region, it mixes some of the bottom sediments and rips up some of the macrophytes and pushes them along the shore. Then they can start to decompose, and I think it's that decomposition of that organic matter which then provides the nutrients to then formulate a bloom. But it's a sequence of a number of these meteorological events and water quality parameters I've been measuring. That's probably key here. But unfortunately, it doesn't happen all the time. So you can't use it for a predictive tool.
Would the sun angle have an effect on one side of the lake or other, seasonal angle?
Yes.
Okay. Maybe there's an ag person, they might be able to comment on that alternative feed for dairy cows. And if there's somebody that maybe Ron O'Rell or
this is Kirsten from Pro Dairy at Cornell University. There's lots of work being done on sort of whole farm mass nutrient balances, and we can sort of dial in the diet of the cow to maximize nutrition to the cow, but reduce, excess nutrients coming out in the way. So that can be as high, like fancy or low tech, as you can imagine. But mostly we can sort of adjust the inputs to the ration. And truly the best way to sort of dial that in is to maximize forage production on the farm. And then, really dial in those other imported feeds to the cows. But if there's any nutritionists on the call or dairy farmers who want to add to that, that'd be great. But we can sort of look at that and are already paying attention to it.
Thanks, Kirsten.
Good to hear. Last question, does the SWOT model take into account the BMPs that are currently being used when attributing?
From what I gleaned at the 9E plan meeting where this was first presented to me, Todd Walters from Cornell worked really closely with soil and water to try to figure out where fields are actually using BMPs and where fields aren't, where the spreading's actually happening, where spreading's not happening. So there was a pretty tight connection between those two groups.
One last part of that same question, does heavy rainfall events, we are seeing the effect of legacy phosphorus as far as nutrient loading?
It can be, because clearly you're going to erode some of the soil that's on tree banks and other things, and that phosphorus has been there for many, many years.
Okay, well, we need to move on to our next presenter, but it's been very informative, Dr. Halfman. Thank you very much. And
thanks, guys.
Appreciate it. Again, folks use the chat method to put questions to. And before we get into, I want to Forgot to thank my co-host, Dave Carr, our IT expert, the fellow that's let you in from the waiting room and is putting up these, helping with all of this Zoom call. So thank you, Dave. And next up is